The first one is the messenger RNA. Messenger RNA consists of single strand of variable length. Its length depends upon the size of the gene as well as the protein, because we know that mRNA is composed from DNA, and now it would be translated into protein because it is taking the message from the DNA and now transferring it into protein. So mRNA size can be estimated from the size of the DNA and the protein. And for example, a protein molecule is composed of 100 amino acids, and then mRNA will have length of 300 nucleotides. It will have length of 300 nucleotides, because we know that one amino acid is formed by triplet of nucleotide. It is formed by 3 nucleotides, right? So we can say that here 3 nucleotides form one codon or one single amino acid. So if there are 300 nucleotides in mRNA, then they would be translated into a protein, and they form a protein composed of 100 amino acids. So we have to divide the nucleotides by 3, then it results in 100. So there would be 100 amino acids in protein. Messenger RNA is almost 3 to 4% of total RNA in the cell. Messenger RNA is the one that takes the genetic message from the nucleus and then move out from the nucleus into the cytoplasm and then to the ribosome to form a particular protein. This is how a protein is formed, that mRNA moves out from the nucleus into the cytoplasm to form protein. And we know that mRNA is transcribed from DNA. So the base sequence of mRNA would be according to the base sequence of DNA. And then messenger RNA attached to the ribosome, and then amino acids attach one by one to form a polypeptide chain, and then this is how a protein would be formed according to the base sequence of messenger RNA. Next is ribosomal RNA. Ribosomal RNA is a part of ribosome. It is a component of ribosome. Ribosome consists of ribosomal RNA. Ribosome consists of ribosomal RNA plus protein. So ribosomal RNA is transcribed by the genes, or you can say DNA, which is present on the chromosome. It is called ribosomal RNA because it becomes the part of the ribosome. And ribosomal RNA is further packaged with variety of proteins because here it becomes a part of ribosome. And this ribosomal RNA have largest size among the RNA. So 80% of total RNA is ribosomal RNA, and this ribosomal RNA is similar in plants, animals and bacteria. So ribosome is a site where the protein synthesis takes place, and ribosomal RNA is a part of that ribosome. So ribosomal RNA provides the platform for the protein synthesis. Next is the transfer RNA. This is a transfer RNA molecule. It consists of almost 75 to 90 nucleotides. Transfer RNA is a single-stranded molecule, but here it shows duplex molecule at some regions where the complementary bases are present, where the complementary bases bond to each other, and here duplex would be formed. It is a flat clover leaf-like shape in 2D view. So you have to remember that 5' end always terminate at guanine, and 3' end always terminate at ACC. This is one point. And amino acid will be attached to the transfer RNA at the end. You can see that this is amino acid leucine. It would be attached to the transfer RNA at the end. Here you can see that this transfer RNA is composed of 3 loops: theta loop, D loop, and this middle loop. Middle loop is composed of 7 bases, and the 3 form the anticodon. So here's the MCQ that you should know that anticodon is present in transfer RNA, and the codon molecules are present on messenger RNA. For example, anticodon is GAA, and the codon is what would be the codon? Guanine always pair up with cytosine, adenine with a thymine. So this CTT would be present. This is the codon, and it would be present on the messenger RNA, and anticodon, which is GAA, it would be present on transfer RNA. And when they both attach to each other, they form the bond, they link to each other, and this is how an amino acid leucine, it is carried by the transfer RNA. This T loop recognize the activation enzyme, and theta loop recognize the specific place for the ribosome for binding, because during the process of translation, when the transfer RNA is involved in forming protein synthesis, here the ribosome subunits are also present. So theta loop is the loop that recognize the specific place on ribosome for binding. And there is one transfer RNA molecule for each of 20 amino acid, and 60 transfer RNAs have been found, and human cells almost contain 45 different kinds of transfer RNA molecules. So these transfer RNA molecule transport a specific amino acid from cytoplasm to the surface of ribosome for the synthesis of protein. So here this amino acid chain would be transferred from the cytoplasm to ribosome for protein synthesis. Molecules. Conjugated molecules are those molecules which are composed of two different kinds of molecules. So it includes glycolipids, glycoproteins, lipoproteins, and nucleoproteins. So here you can see that glycolipid is composed of carbohydrate and lipid portion. So these are two different kinds of molecules which combine with each other, which make a connection with each other and form a conjugated molecule, which is glycolipid. And the carbohydrates, this carbohydrate have polar head. They have polar head. And glycolipids are present in white matter of the brain and myelin sheath as well. You know, myelin sheath in nerve fibers. Here glycolipids are also present. And even in the chloroplast membrane, which is in plants, we found glycolipids in chloroplast membrane as well. Next is the glycoproteins. Here the proteins and carbohydrates combine or attach to form glycoproteins. So this is a new kind of conjugated molecule, and they are attached covalently. By the covalent bond, glycoproteins are widely distributed in the cell because these glycoproteins act as hormones, they act as signaling molecules, they act as transport proteins. The proteins which act as transporter, they also act as structural proteins and receptor as well. Even the blood group antigens are also glycoproteins, and these glycoproteins have an important role in blood grouping. Next is the lipoprotein. Lipoprotein is composed of lipid and protein, and its example is phospholipid. Phospholipid protein, it is widely distributed in plants and animal material, and it also occurs in milk, blood, nucleus, egg yolk membrane, and even chloroplast. Next is nucleoproteins. Nucleoproteins are the protein which are composed of nucleic acid and proteins. We know that nucleic acids are DNA and RNA. So when they bind with the proteins, they form a new kind of molecule which is nucleoprotein. And nucleoproteins are also found in the chromosomes and ribosomes. For example, DNA bound with a histone protein, so it is also an example of nucleoprotein.